Let's go back a couple of steps now and talk about how the overexpressed FGF affects the mitochondria's release of cytochrome C. FGF binds to a receptor tyrosine kinase here, and this activates the receptor tyrosine kinase, causing it to cross-phosphorylize itself, and in phosphorylating itself, it recruits another protein, PI3 kinase. This becomes activated when around the phosphorylated receptor tyrosine kinase, and it phosphorylates a PIP2 phospholipid head to a PIP3. So this is now 3 phosphates instead of 2. This PIP3 then can bind to PDK1 or AKT, and we'll see in the next way that the AKT and the PDK both bind to these PIP3s, and PDK1 and the protein mTOR will phosphorylate and activate the AKT protein here, which is held in place by the PIP3 until when it becomes activated through the mTOR and the PDK activity, it dissociates. And in dissociating, the activated ACT will then act as kinase and phosphorylate BAD, so that BAD becomes phosphorylated and it binds to a protein called 14-3-3. And also it releases another protein, the apoptotic inhibitory protein, which becomes active. This active protein will then inhibit apoptosis, and we will get continued proliferation of the cell, and it will continue to grow. And this is important because if there is an issue with this pathway in any way, it can be detrimental to our ability to do apoptosis, right? And that's what we're going to see with the P10 mutation, where we'll talk about later, but it does cause it so the cells are not able to go in apoptosis, and they can become more cancerous than they were, and they're not able to die.